Hey everyone, welcome to topic 3, fertilization and development. So, in topic 2, we discussed a lot of the hormones, a lot of the processes that lead up to the point of possible fertilization. So where we're going with in topic 3 is we're going to look at the physical process of fertilization and then that development from that point of fertilization. So we're going to look at reproductive technologies, fertilization, cleavage, gastrulation, and implantation, neurulation and organ development, supportive structures, so your placenta, amniotic cavity, stages of fetal development, environmental and fetal development, and partition and lactation, so another role of 2 of your hormones. So, the first process we are going to talk about is fertilization. So fertilization is the joining of male and female gametes, in other words, an egg and a sperm, to form a zygote, which by definition is a fertilized egg. So what happens is an egg will have 23 chromosomes, a sperm will have 23 chromosomes, and after that process of fertilization occurs, that zygote will then have 46 chromosomes in total. So if we look at this diagram, this diagram is showing you that you have your one egg and then you have quite a few number of sperm. We look at the size difference, and I know we talked about this in one of the earlier topics, but an egg will contain more cytoplasm than a sperm. So, occurs within the oviduct, unique coating layers of the egg ensure that only one sperm may successfully attach. So once this one sperm joins and fertilizes with that egg, it then allows there to be that unique coating that only allows one sperm to fertilize. Now, I know there are situations where we talk about twins, etc. We will discuss that later, but what we need to look at is an egg has 23 chromosomes, a sperm has 23 chromosomes, and the max number of chromosomes in a human cell is 46. So if we end up with 2 eggs and 1 sperm, we're now in a situation where we have too many chromosomes, which is not good either. Okay, so how this process works. Sperm cells release enzymes from their acrosome cap, so this is at the head of the sperm, to digest the outer coating of the egg. Sperm cells move farther through the coating towards the plasma membrane. Okay, so if we look over at our diagram here, we have a few sperm, okay, but what we are looking at is, if we look at sperm D, this is the one that is going to start penetrating through that outer layer of that egg, which is trying to get on the inside of this egg. Which leads us to number 3. Once a sperm enters the egg's plasma membrane depolarizes. So this goes back to our Unit A when we talked about the nervous system. So we talk about depolarization. We don't necessarily need to know the process because it is kind of complicated, but we need to understand is that there is that depolarization that is occurring, and that depolarization is going to prevent other sperm from entering. So once we are there, the nuclei of the sperm and egg fuse, resulting in a single cell with a complete set of 46 chromosomes, which we refer to as a zygote. So what I want you guys to have a look at is, I did post a few YouTube videos that go through a lot of this development process. It is important that you look at those. They do give you that visual. They do give you that physical process, and they are very, very beneficial. Okay, so once we get into a zygote, so fertilization has occurred, we've taken 2 cells that have had 23 chromosomes, both of our sex gametes, and we've now combined them into one, which has 46 chromosomes. Once we are here, we are now going to go through the process of taking that single cell, that egg and the sperm, which have united into that zygote, and we're going to go through a process in which in the next 9 or so months that that zygote will then start to differentiate and will turn into a fetus. So early on, the first kind of once after we get our zygote, we are going to go through the process of cleavage and implantation. After fertilization, the zygote continues moving through the oviduct towards the uterus. Remember, the oviduct we refer to also as the fallopian tube. Okay, so once fertilization occurs, that zygote is going to move down through the fallopian tube into the uterus. During this time, several divisions occur, leading to the formation of a morula. So this is a really, really good visual for us. We start with a 2-cell zygote, turns into 4, turns into an 8-cell zygote, and then we are going to end up with a structure called a morula. What we need to remember, and this is going to lead us into Unit C very nicely, is this process is called mitosis. Mitosis is the production where one cell will reproduce 2 exact same cells. So this is what's occurring here, is that we end up with a 2-cell zygote, it doubles, we get 4, it doubles, we get 8, etc. and etc. So the initial cell division occurs rapidly, leaving little time for cell growth. This process of cell division without enlargement is called cleavage. Over the next 3 to 5 day period required to reach the uterus, cleavage occurs several times to form a sphere of 16 cells. This is the morula. So realizing that morula now has 16 cells. So remember that once fertilization occurs, going back, we're in the fallopian tube, that group of cells is going to then migrate towards the uterus. And when we get here, we end up with a structure called the morula. So now, we've ended up with this morula, this group of cells that has gone through a mitotic division, and it's now ended up in the uterus. So what happens is, from a morula, it's going to develop into a blastocyst. That's an important word. A fluid-filled structure consisting of 2 layers, the trophioblast and the inner cell mass. That is important for us to understand because these certain structures, the inner layer and the outer layer, are going to have a different role as we move forward in this development. So, between 5 and 7 days, the blastocyst will begin attaching itself to the endometrium. This is known as implantation. So what we need to remember is we've left the fallopian tube, we've entered the uterus, okay? And inside the uterus, we know that we have an inner layer called the endometrium. And this is where this ball of cells is going to implant on, which we know when we go back, as we just talked with the menstrual cycle, is that we don't want that endometrium to be shed. So this is where, in case it's going to deal with our hormones to try and keep that endometrium in place because implantation has occurred. Implantation is complete by days 10 to 7. If successful, the woman is now said to be pregnant. So when we start to look at what happens from a hormonal level, we know that when we looked at the menstrual cycle, we had a fluctuation in a lot of our hormones. They went up, they went down. But this is where, okay, things will start to hormonally look very different in a female because we are now having our hormones try and keep that endometrium in place, which when we talked about in topic 2, we talked about our HCG hormones, that pregnancy hormones that's detected. So when we look at that, we now can start to see why women in their early stages of their pregnancy will start to feel sickness. They start to feel different because they have a surge of hormones that weren't necessarily there before. So leading into post-implantation. So post-implantation is referring to that group of cells have now embedded itself into that endometrium wall, but that trophoblast begins secreting a hormone, the human carotid gonadotropin, the HCG. This is known as the pregnancy hormone. So when a female takes a pregnancy test, this is the hormone that is being detected to indicate if that female is pregnant or not. So HCG maintains the corpus luteum, which has a very, very important role, because we know that if someone is not pregnant, that corpus luteum will produce progesterone and estrogen, but then it'll start to drop and decrease. But what happens is that corpus luteum is maintained, which allows your body to keep secreting your estrogen and your progesterone. What that allows it to do, and if we go back to the function of our hormones, is that it's going to allow our body to maintain the endometrium and will prevent menstruation. If menstruation occurs, we are going to lose that endometrium, which is not good because this ball of cells have now implanted itself into the endometrium. That is why our body needs to keep secreting that estrogen and progesterone to keep that endometrium intact. What happens is your HCG levels decline after the 2nd month of pregnancy because you have the placenta, which is able to secrete its own hormones. So that what your placenta will be is your placenta is the gateway between that potential fetus and the mom. So until that is fully developed, we need that source of hormones somewhere to keep that endometrium intact, which is why that HCG is going to be present. Okay, so after we've kind of gotten past that past week, we're going to lead into week 2, which we refer to as the process of gastrulation, where we've gone from a zygote. The zygote is turned into a blastocyst, and now that blastocyst is going to start to change. This is what we refer to as that blastocyst is going to go through a process of differentiation, which is going to allow this group of very general cells to start to differentiate into cells that will then turn into skin, that will turn into muscles, that will turn into organs, etc., etc. So looking at this diagram, very good diagram. So we have this blastocyst that'll turn into a blastula, that'll turn into a gastrula. But what we look over here, we have 3 layers, which are very, very important. We have the endoderm, which is referring to that inner layer. Our mesoderm, which we are looking at as a middle. And we have our ectoderm, which is that outer layer. So a space known as the amniotic cavity forms between the inner cells and the tropioblast. This is where the baby will develop. The inner cell mass flattens into 3 distinct layers: the outer ectoderm, inner endoderm, and middle mesoderm. This is the process known as gastrulation, after which the blastocyst is referred to a gastrula. I know there's a lot of big complex words, but I will go through it again. Don't worry, you guys, this is not that bad. But what we really need to take out of this is that we've now started from a group of cells and we are going to differentiate into those 3 layers. So each of the 3 layers is referred to as a germ cell. And this is where the really, really important word differentiate comes into. They're going to start to specialize. They're general cells, and now they're going to start to turn into cells that have very different roles from each other. So the ectoderm, okay, is our skin, our hair, nails, nervous tissue, sensory organs, pituitary gland, and tooth enamel. If a cell was part of the mesoderm, it has the potential, this is the important thing, it has the potential to turn into blood vessels, muscle tissue, bone, cartilage, blood, heart, kidneys, internal reproductive organs. And then we look at our endoderm, which is the inner layer, which then will have the potential to turn into the respiratory tract, digestive tract, gallbladder, pancreas, and your thyroid gland. Okay, so now we've kind of taken the first 2 weeks of development, and remember, we're just going through the overall general process. We don't need to get hung up on the very, very small details. What we need to know at a Bio30 is what are the general processes. So we've now kind of entered the end of week 2. We're going into week 3, where we've had differentiation occur. We have these 3 layers of germ cells that have the potential to turn into these specific cells. So what we are going to start to see is in weeks 3 to 8, okay, the germ layers are going to begin to fold and early organ systems take place. So it's really important that we kind of look at the general timeline. We are only looking at week 3. This is where we are starting. So this is still very, very early on, because we generalize a pregnancy is 9 months, but when we look at it from a term of weeks, a pregnancy is going to last 40 weeks, 40. So what we are looking at is we are now kind of focusing on weeks 3 to 8. 3 to 8 is probably the biggest, most important when we look in terms of development of specifics. Okay, by 8 weeks, majority, and I say majority because not all, but majority of the major life functions will be developed and differentiated by then. So when we look at week 3, we have the formation of the notochord. This is going to develop into the brain, spinal cord, and this is the process of neurulation. Also, a primitive heart will start to beat as early as this beginning of week 3. One of the things that pregnant women are told to take is folic acid. Folic acid, if we've heard of that, has been linked to your development of the neurulation, the spinal cord, etc. Week 4, formation of blood cells. Lungs and kidneys will start to take shape, and small buds which develop into arms and legs will start to appear. A distinct head is visible. Week 5, eyes open, cells of the brain undergo rapid differentiation. So that word differentiation just is meaning that they're going to start to take on different roles than their neighbor cell. So we know that when we look at a brain, not every part of the brain is responsible for a certain thing. So maybe they'll start to differentiate into the occipital lobe, the temporal lobe, and then they'll start to take shape into those specific roles. Week 6, we are continued development of the brain, lengthening of limbs. By week 7, organs and skeleton are almost completely formed. External genitalia begin to differentiate. And we talked about this because we know that from topic 2, when we talked about our hormones, at week 7, we start to see if there is testosterone. That's when the male genitalia will start to be produced. Nervous system begins to coordinate body activity. And by week 8, the embryo is the size of a paperclip. 90% of the organs are established. So we're at week 8 out of 40, and we have majority of our organs that have been established. They are not fully functioning, but they have been able to differentiate from those different germ layers to turn into those specific organs that are required. Okay, so we're going to focus on a little bit more specifically when we talk about the development of the nervous system. But the first word, neurulation. The folding of germ cells to form the notochord, which will develop into the brain or spinal cord. So in order to form the notochord or the neural tube to form properly, and this is where I mentioned, guys, is your folic acid. Okay, if there is not enough folic acid at this point, this can lead to some issues. Specifically, the folic acid is linked to spina bifida, and you guys can do a little bit more research on that. That was just a little bit of a bonus information. Okay, so on to structures that support the embryo. So now we have an embryo formed. A lot of that differentiation occurs. What we are going to look at here are just a few things. Your alliantosis. This forms the foundation for the umbilical cord. So if we look right here, this is where your alliantosis is going to start. Your alliantosis is the umbilical cord. It's going to attach to the placenta, and that placenta is going to attach to mom. We have a yolk sac, which is going to form our digestive tract, but that is going to shrink after 8 weeks. This one, the placenta. The placenta attaches to the fetus, to the uterine wall, where your metabolism exchange occurs. This is composed of embryonic and maternal blood and vessels. So your placenta is that gateway. That is how that embryo fetus will always get the required nutrients. But it is very important that we understand that the placenta, that the mom and baby, they do not share blood. There is an exchange of nutrients, waste, gases, but they do not physically share blood. Your amnion encloses the fluid-filled sac, which protects the embryo during development. So your amnion, when you hear that in labor that a woman's water breaks, it's the amnion sac that is going to rupture. So that is what breaks, that allows that amniotic fluid to be released. Okay. And then we have our chorion layer. So our chorion layer forms the fetal portion of the placenta. So briefly, if we go over this, we just need to understand the functions of the placenta. So we've gone through the nutritional functions, transports nutrients from mother to fetus. It's going to store your nutrients. Okay. It's going to excrete. So it's going to transport wastes. Okay. Respiratory, we need to look at oxygen and the carbon dioxide from the fetus to the mother. Endocrine, we know that hormones are secreted through the blood. So how do those hormones pass through mom? Well, they're going to have to go through the placenta. Allows hormones from the fetus to diffuse into the mother's blood. Okay. And immune functions, transports antibodies from the mother to fetus, so that if mom is sick, mom has a cold, the fetus is not going to be majorly impacted, hopefully, because of that exchange of those antibodies. Now, what we need to look at is the placenta does not filter out harmful substances. So your body, a female's body, does not have the ability to say, oh, that's not good. It's when we look at things like drugs, alcohol, etc., your body does not have the ability to filter that. The placenta is going to still allow that alcohol, the nicotine, the drugs, whatever is taken, that to enter into that fetus. So, and we don't always have to look at the illegal drugs as well. We can look at things like your pharmaceuticals, taking cold medication. And a lot of the times when a female is pregnant, they are very cautious. They have rankings. And if you actually ever go to a pharmacist, they'll tell you that there's different rankings of how drugs are coded and how they could impact fetal development. Okay, this just shows exposure to tetragens. Tetragens is the word that we talk about, those bad things that can come across. Okay, I'm not really sure what that animal is. Or flowers, etc. Okay. Last kind of part of topic 3 is relating specifically to our fetal development. So we know that we've gone over specifically weeks 1 through 8-ish. That is where the biggest chunk of information and what is expected out of Bio30 for you to know is that week 1 through 8. But what we're going to look at is we're going to look at generalizing the rest of that pregnancy that leads to that 40 weeks. So we're going to look at the first trimester, second trimester, and third trimester. So when we look at this, it's really important, guys, that you need to know weeks 1 through 8, which we've gone through in detail. But when we look at our first trimester, okay, week 1 through 8, we've gone through in detail. But what's going to happen is your body is going to, the baby's body will start to grow, skeletons begin to harden, and reproductive organs become distinguishable. So when we look at the finding out the gender of a fetus, that really doesn't happen until probably 18-ish weeks, okay? And that's because you have to wait for the development of those external reproductive genitalia to form. So when we look at first trimester, it's down here. So we're looking at weeks 1 through 12. Now, moving on to our second trimester, weeks 13 through 24. So this is the point where our heartbeat is strong enough to hear with a stethoscope, rapid brain growth, nervous system begins to function, okay, not develop because it's already been developed, but it's starting to become more functional. Mother begins to feel movement. Fetus will not likely survive if it was born at this stage. So that's the other thing we look at when we talk about premature babies. We talk about babies being born before preterm, okay? And this is because depending on what week, different things develop at different times. And yes, it has been formed, but maybe those lungs aren't strong enough, etc. Okay, third trimester, weeks 25 through 38. I know it says 38. Most pregnancies do go to 40, but if a baby is born at 38 weeks, it is still considered to go to full term because your structures are all developed enough. But when we look at this, a third trimester, this is where brain development, organ maturation will occur, and the fetus will normally double in size. Okay, so when we look at delivery of the baby, the onset of labor or uterine contractions is regulated by positive feedback. So this relates back to our endocrine system. I told you your hormones were not going away. So when we look at this, your cervix dilates, and before we left, we know that we talk about this is where your cervix is from 0 centimeters dilated, and it's going to go all the way up to 10. When the cervix is fully 10 centimeters dilated, that means that active labor, I guess not active labor, well, I guess active delivery is occurring. So once the cervix starts to dilate, you get oxytocin released from the pituitary. Uterine muscles will contract. So this is where a female will get those contractions. The closer the contractions they are, the more, I guess, the closer it is to delivery. And this is where a fetus is pushed downwards, causing the cervix to stretch. Okay, so this is in a standard typical labor. And when we look at things, we talk about women having to go for emergency C-sections, etc. Is if something goes wrong in someone's birth plan, you then have the ability, like if baby cannot come out, that's when they'll go to the OR, a C-section, where they'll have to cut through the abdomen wall, cut through the amniotic sac, and then remove baby that way. Okay, hormonal control of lactation, we did briefly talk about this before we left, but high levels of estrogen and progesterone suppress the production of milk, so that's during your pregnancy. But after, the pituitary is going to release prolactin, which will then stimulate your breast milk. The actual release of the milk from the breast or lactation is mediated by oxytocin, which will cause the contractions, okay, which then helps with the physical release. Prolactin, both of those for your lactation, okay? So this video, I'll post a couple of videos of kind of going through the fetal development. They're kind of cool to watch, you guys, so I really suggest you watching them. Whew. We're done. Okay, so that was a brief overview of topic 3. I know I talked a little bit today, longer than I normally do, but hopefully that was okay for you guys.